So this is the last lecture today. It is a long day. Right, so now we're going to talk about efficient strategies to find the area of interest and tiling and autofocus. So all these things are really important. I think throughout what we've done so far, um, we spoke a lot about the way the point spread function is formed and the light behaves and bleeds through. And then later we spoke a lot about the different hardware. So I hope that you feel comfortable with your microscope and how it works now. And today there was a lot about the objectives. And we have seen that one of the things that can be limiting with the objectives, especially the high resolution objective, is the field of view. And so we need to develop some sort of strategies to be efficient. And when you come to me for the student imaging workshop, I will show you how easy it is to use the automation of our microscope to navigate in the sample. And I think this year we have no one who has a manual stage, if I am not mistaken. I think everybody has an automated system, so that is very good. Right, so tiling. I think sometimes people call it mosaic, overview, tiling, and Nikon calls it large image. Does anyone have a Leica system and can tell me what this is called for Leica? Or an Olympus system?
I we use a Leica system in the lab and we usually call it tiling, but
Okay,
I
tiling. So maybe
don't remember how it is called exactly in the software, but we always called it tiling in the lab.
right, maybe it is called tiling. So it's quite yeah, in uh, different companies call it differently, but all this is exactly the same thing. And this is a very nice tool. So basically, when we want to tile, what we are doing effectively is that we're increasing the field of view. So we are taking lots of images and putting them next to each other and then fusing the whole thing so that we have one field of view and then we can image all this. This is live samples that has been imaged that way. But that means that we have a lot more cells, a lot more of the tissue, and we can get good statistics. Now, we do not have to do this only for live samples, clearly, but we can do this for fixed samples as well. And it is an excellent way to navigate in your sample instead of finding your sample with the eyepiece, looking at fluorescence, bleach, bleach, bleach. Oh, nice here. I move here. Oh, nice here. Oh, have I been there? Yeah, I go back to the same place. No, we don't do that. What you do is that you find your sample and then you tailor the microscope to take a big overview of your sample. And clearly, if you use a single point confocal, this is going to be slow, but we don't have to be slow because we are going to see this next week. You can use very big pixels. You use a low magnification objective, you get big pixels, you get the full field of view, and you image with the maximum speed, no averaging, whatever. You can go very fast as well and image the whole thing. Once you have your whole sample like this, an image of your whole sample, you can use, and this is true for all microscopes that have an automated stage, you can tell the machine to go here and move the stage here. And then you can record the position there. So once you have the overview, you can record these positions that you're interested in and log them in the software, and then tell the software, image these two positions one after the next. And in between, you change all the settings so you have the settings that you want. So you use, for example, low magnification, the minimum number of channels so that you can identify the areas of interest, no averaging, full field of view, and you save this image as a reference so that you can show the context of the positions that you're interested in. Then afterwards, you change all the settings, maybe a different objective, and you use so this is on a single point confocal, you would use very large pixels to acquire this type of images, a very big pinhole so that you can lower the laser and get a lot of light anyway. You can use bidirectional scans. So all these type of things we will talk about later in the course. A resonance scanner if your machine has one, and then a very fast speed. And then camera-based system, you can use a low illumination, even if the images are noisy, as long as it shows you the areas of interest, fine. And then you can bin as well. So all these type of parameters we will talk about later in the course. But it is totally possible to image the whole sample quite quickly. And then you go back and you change objective and you do a Z stack. You have all the colors that you're interested in and the correct pixel size and all that type of things so that you have low noise. And so this is really a very efficient way to navigate in your sample, and you're sure that you are not imaging twice the same place and we're not bleaching. Jessie, you have a question.
But if we have inverted microscopes, when we if we want to change objective and you need to change a medium between different objective, you move the slide and then when you put it back, you are not exactly in the same position anymore.
That's true. We have tricks.
Okay,
Yes.
wonderful.
There's lots of tricks. So I can show you my tricks when we have your workshop. But basically, there are several ways to do that. So if you are, so of course, when you would take the big overview, you're going to use like an air objective. But if you want then afterwards to image with an oil objective, for example, there are several ways of doing it. One of them is depending on the stage insert that you have. You can try to push your slide into one corner to make sure that you come back more or less at the same position. Might not be exactly, but it's going to be quite close. You get used to it and then you do it. It's actually quite reproducible. But it will not be exactly the same, but it will be very close so you can find the position of interest. That is one trick. And the other trick is if you have a stage insert where you can adjust so you put the slide on the stage insert, and some of them you can actually adjust the width. If you have something like this, you have holes around, and all through the hole there you can put the oil. So I will show you this when it's your turn. Jessie, where are you located? I can't remember.
Like when you mean?
Where are you?
I'm in Malaga, in the south of
Spain. Yeah, you're not in Stockholm. So you're going to be remote. But I can ask Gabby to sort of film, and then I can send it to you.
Wonderful. I'm very interested.
Yes. It works very well. We do it all the time. Yes. So all microscopes that have an automated stage can do this, and you will see during the workshop how powerful it is. This is a great way to navigate in your sample in an efficient way. Another way to be very efficient is if, for example, you're looking for a needle in a haystack. So you have, for example, a big culture and you're looking for only the cells that are dividing. Or you're looking, in this case, for a certain combination of cells next to each other. You can do something called smart microscopy. Nowadays, all manufacturers, like the big four manufacturers, definitely, I mean Nikon, Zeiss, Olympus, and Leica, they all have something called smart microscopy. Now, everybody calls it differently. Obviously, they cannot agree on the name. So in Nikon, it's called Jobs. And I think in Zeiss, what is it called? Anyone knows? Is it something experiment? I'm not sure if, but let me explain to you what it is. Maybe it comes back to your head. So this is an example of smart microscopy. We're taking images, fast images. We acquire a big area. In this case, it was a multi-well plate, and we acquire a big area with. It's noisy. It doesn't matter because the resolution in here is sufficient for us to identify the objects that are potentially interesting. So we do this very big scan of the plate at low resolution and very fast, and then we do a first round of image analysis. And when we do that, we identify the potential pairs. In here, we're looking at yeast, and we were only interested in the type of yeast that were now with this red stuff. Okay, so we had a mother and a daughter cell next to each other, whereas the others, it's not sure. This one is probably two daughter cells. This one has no daughter, etc., etc. So they are excluded, the others, and so we analyze, we identify which pairs we want to analyze, and then the software automatically writes down a list of all these positions, and then it changes automatically all the settings to whatever you have decided that works for these for your real analysis, and then it re-images everything, only the stuff that are potentially interesting to your analysis. So in this case, this is a yeast plate. You have so many yeast, and most of them are actually not potentially interesting. So using this system of smart macroscopy, you only get the images that you're interested in. It's pretty cool. So so you can go for a coffee and while the whole thing is working for you. So smart macroscopy is a very nice way to work as well with the automation of the macroscope. Autofocus is also very, very important, clearly, for the automation. When you and one thinks about autofocus, like, okay, it's live samples, and over 10 hours, the focus is going to drift, so we need to have an autofocus. This is not the case at all. Most of the time, we use autofocus with fixed samples. There are many reasons why the focus could go wrong, and one of them is, for example, in this piece of skin. It's a long and slim sample, and from one end to, if you focus in here and you move to the other side of the sample, it's not going to be in focus. So we have to have a strategy to keep the focus throughout all our imaging, either in time or in space. So one side of the overview here is not in focus, and in here we have a problem of focus even within one field of view. So you can see one image we have here, it's in focus, and here it's not in focus. So there are reasons why things can go that way, and it makes it much, much, much harder to analyze these images. And clearly, if you're using a single point confocal and you have a long sample like this, you don't want to have to do a Z-stack to compensate for the focus, when really you could use an autofocus and be in focus everywhere and skip the Z-stack. So this type of things, it's difficult to analyze, and you get no reliable intensities either. So let's see what we can do. What we need to define a focus strategy to allow us to get reproducible data. And because clearly, if you have a sample like this, and one day you focus in here, and the next sample you focus in here, it's not very reproducible, right? So the two questions that we want to answer to get our focus strategy are: how do we find the focus reproducibly between sample? So I take my sample, I focus. Then I take another sample, I focus. Yes. How do I know that I'm focusing in the same way, so that my data is actually reproducible between samples? But also, how do I maintain the focus during the imaging? Here, if I image from this side to this side, the focus is going to be different. So this is what we want to solve with an autofocus. So I will show you now what we can do with autofocus. So the applications for autofocus can be a time lapse with time. Clearly, the machine moves, so you have to compensate. But it can be also when you're imaging a cold sample with an oil objective. If you take a sample straight from the fridge and you image it with an oil objective, the focus will drift very fast, and it will drift for a very long time. So you may think that after 10 minutes, your sample is at room temperature, and you may think that the oil is at room temperature, but this is not the case, because we're imaging through the oil, and the oil also warms up. So there is a difference in temperature between your sample, especially if it comes from the fridge, and the oil, and that creates a lot of focus drift, and that lasts for a very long time. So I'm talking about like 1 hour. So that can be compensated by autofocus as well. And also, so for example, you can imagine that if you do a slow Z-stack, and then the focus is moving at the same time as you do the Z-stack, it's going to be an issue. And then we have, when we image over long distances, for example, if we do an overview, if you image a multi-well plate or something like this, or if we have a long sample, then we clearly want to use an autofocus. Another application would be when we have cells at the surface of a gel, and the gel is never flat, so we would want also to have an autofocus there. The major causes of focus are temperature drift, or the sample is not flat. So this is very common. It can be because you didn't do a good job at mounting your sample and your cover slip is sideways. Or you didn't put your samples flat on the stage. So that can also happen. Or the insert is not flat on the stage. So there's quite a number of things like this that makes the sample be slanted. And then you're going to have a difference from one end to another. So one typical example that Gabby will talk about on Monday is that if you put your cover slip flush on the edge of the slide, you are likely to have some problems on an inverted system, but she will explain why and where you should put the cover slip on your slide. See, there's a lot of details that we have to think about when we do microscopy. It could also be that you're using one of these optical plastic bottom, for example, multi-well plate with optical plastic. And yes, they are nice and they are cheap, but they are often not flat. So that is the problem. And between manufacturers and even between lots of multi-well plates, there's a huge variability, unfortunately. So one of the main issue we have with these optical plastic bottoms is that the bottom is not flat. So it is very important to keep the sample exactly at the same focal plane. This is critical for image analysis and for the reproducibility. So we want reliable intensity information. As you know, where fluorescence, when you're a bit out of focus, the fluorescence just goes all the rings, right? What you're imaging is the rings of the point spread function. You're not imaging the big blob. So that is a problem if for the reliability of the intensity and also the reliability of the shape. So image analysis does not like autofocus things. The solutions are hardware or focus, autofocus, and I'm going to explain to you what it is, and these can be combined. Or if you have a problem with the focus within one field of view, like I've shown you before, where the center is in focus and the quarter, for example, or one side is not in focus, then what you need to do is reduce the field of view so that everything is in focus. And otherwise, clearly you can add a small Z stack, compensate and do a maximum intensity projection, but that takes more time, more imaging time and more bleaching, of course. So I'm going to introduce now hardware and software autofocus. Software autofocus. This is the microscope is going to take several images of your sample across. It's going to do basically a little Z stack. Okay, so we have here Z1, Z2, etc. So it does a little Z stack and then it analyzes the images and tries to find the image that has the best contrast and considers that this is the plane that we want to image, and then it goes there. So this is how a software autofocus works. It does a Z stack and acquires images. So this works very well if you use low numerical aperture objectives, because it's it has a big depth of field. So we have there's not a lot of difference when you move a little bit. So that works quite all right. But there are disadvantages. So first, it images your sample, so it bleaches your sample. Also, it can be quite time consuming. If you want a fine autofocus, it can take quite a lot of time. So it's not something that if you image a multi-well plate, you wouldn't be able to do a software autofocus on each tile, for example. And also, if the contrast is dim, since it relies on the contrast in the image, if the contrast is dim or you have a low signal, it doesn't work because it's not going to find anything. And of course, if your sample is a little bit in 3D, for example, if you're imaging vesicles that are everywhere in the cell with a high NA objective, there are several planes that would be in focus. And then clearly that through the software autofocus, it will not be able to find which one is the best plane. So the advantages of hardware autofocus is that it is available on all microscopes, inverted and upright. And then this is the second type of autofocus. It's a hardware autofocus. So again, different companies call things differently, but the principle is very, very similar. So here we have the objective. This is an inverted system. The objective is below the stage. And here we have the cells, and we have here the cover slip. And what happens is that we're focusing in here on the cell. So here they say there's a focus drift, that can be temperature, that can be anything. And then what happens is that now we are focused, the focus level, the cover slip has dropped basically. So we're not focused at the same place. We want to compensate for this. So we have a light source, which is normally in the high wavelength, like 800 something, so away from the range that we are using. And then this light source comes into the objective and bounces because of reflection. It bounces on the cover slip and it goes back down. And then we have something that detects, we have a little detector that detects this light of a long wavelength and determines the distance between the objective and the cover slip, or the place where it reflects. And so you can set an offset yourself and say, find the surface of the cover slip, always go 3 microns higher. And so then the software moves the objective to the correct Z plane and it stays there. It just measures and goes back to the same. And this does not interfere with your imaging because it uses a very long wavelength. So the disadvantages of this type of hardware autofocus is that it requires a refractive index interface, because the reason why the light reflects on here is because we have refraction. We have a different refractive index here and in the glass, and therefore the light is going to be refracted completely and go back to the sensor. If we have a perfect sample, for example, we have a sample that is in a high refractive index mounting medium, and here we have glass, and here we have an oil objective, so we have oil here, it doesn't work. You must have a refractive index interface, so an imperfect setup for the hardware autofocus to work. So it only works if it also, another thing is that it only works if the focus of interest is at a fixed distance from a cover slip, for example, always 3 microns. So if we have a sample like this, it doesn't work. We can't use this. Like, for example, cells at the surface of an uneven gel, we cannot use it because it always goes back to the same distance, the same offset. And it's not available on upright systems. I think we have one person who uses an upright system. It's not available on an upright system. So the advantages of the hardware autofocus is that it's instantaneous, so it's very fast, and it does not interfere with the imaging. So it goes on in the background without you knowing, and at the same time you can image. And so therefore there is no bleaching at all. And it works for any type of application where the sample is at a fixed distance to the cover slip. Okay, so they have different names, as I mentioned. Nikon calls it the perfect focus system, which is the one that completely throws me compared to PSF and PFS. Thank you, Nikon. And then we have Zeiss calls it definite focus. Leica called it adaptive focus control, AFC, and Olympus says zero drift control. So you might have heard these words about your microscope, and one of the assignments that you will have this weekend is to go back to your microscope and explain to me all the steps, to tell me first if you have some hardware autofocus on your system, because it's an option. So it might be that you have it or that you do not have it. I hope for you, you have it. It's a great tool. And if you have it, then you should describe the steps that are needed for you to set it up. It's very, very fast and super cool. And it works a charm. So I hope that you have one. Now I just want to tell you, if you want to image something that is slanted or complex surfaces. So far, we were just talking about something that is like on a plane. So the plane can be tilted, but it was anyway a plane. Now, if we have complex surfaces, what do we do to stay in focus everywhere? So let's say, for example, we have this is a dish with a gel, and then the gel is a bit sunk at the middle here, and then it's higher on the edges. So we have like a some sort of a concave surface. What do? We can use a trick that we have in the software, and that is not hardware autofocus and not software autofocus, which is that we are going to create an artificial surface by logging many points of focus. And then the software calculates a whole surface. So we go to this position, we focus here on the surface that we want to image, and we log the position, so X, Y, and Z. And then we go to another position, we focus again, so the objective is lower because we are in the center of the dish, and then we log this position, and then we do this again at several places in our dish. And so we set up a matrix of XYZ coordinates like this throughout our dish. And then the software is going to calculate a whole surface based on these points. And so it means that you can go here and it will more or less know where to go in focus. Or you can go here. These are points that you have not logged, but that the software can calculate based on the data that you have given. So the disadvantage of this complex surface imaging, and again, different companies call it differently, is that you have to enter the positions manually. But it works very nicely. For example, if your sample is on the slide and is not straight completely, you just need 3 spots to make a plane that follows your sample. So that is very, very convenient, and that works very nicely. But you have to enter the position manually. And the advantages of it is that it works for uneven surface and sloped surfaces. But it also works for perfect samples with no refractive index interface, because in here we're not acquiring an image, but we're not using the hardware autofocus. Okay. And it works if the sample is not flat. So if you have your sample is a bit slanted or is a complex surface. So it's a nice option as well. It's clearly more time consuming because you have to enter the points manually. And if you have a multi-well plate, forget it. It's not going to work. It only works if you have a few samples. So now I want to just a little conclusion about this this part is that we should use the automation of the microscope to explore the sample and find the area of interest. Make a big overview with very quick settings that allow you to go fast and find the area of interest. And then you log these areas of interest, and then you change all the settings and go back there and re-image. This is a fantastic way to work. And you have actually a lot less risk of being biased than if you look in the eyepiece, because if you look in the eyepiece, it depends which color you're looking at. You're going to be very efficient or not very efficient, whereas the machine is much better than you. So don't trust your eyes, use the automation. So you have less bleaching, you have less, it takes less time, and you have less risk to image the same area twice. So it's all benefit. A reliable focus is critical to get reliable data and a reliable image analysis. So it's very important that you have a focus strategy and you want to answer this question. What will you focus on to start with? So, you have cells. Yeah. What is it that you're focusing on? Are you focusing at the bottom of the cell? Are you focusing on the nucleus? Are you focusing on the top of the cell, on specific molecules? Yeah. Are they always from one sample to the next? Are they always at the same level? Are you at the top of the nucleus or at the middle, at the equator of the nucleus, etc.? So you need to define this. And when you have your focus strategy, you can write it in a paper. And this is reproducible from one researcher to another. So how can you find a way to focus in such a way that your focus position is comparable between different samples? And how will you ensure that the focus is stable during imaging? And again, you don't have to image for hours for the thing to drift. It does drift. And you can focus on the structure that is not affected by your treatment. If you are focusing on the filopodia, for example, to find the area of interest, but you have a treatment that makes the cell rounded, there's no filopodia. What are you going to focus on? So you need to find something that is reliable between your different samples. And also, if you focus on something that is stained in one sample and not in the other, you're going to have a tendency to focus on something else. And that is not reliable. It's not reproducible. So you need to write your focus strategy in the material methods when you write your articles so that everybody can reproduce your data. And if you can use a hardware autofocus, use one. It's a brilliant machine. So you will have to define in one of the, so I'm finished now, but in one of the assignments, the portfolio assignments, you will have to tell us what your focus strategy is. So I am done with this, and I would like to know if you guys have some questions about all this or thoughts. If you have ever tried to figure out how you focus on your sample and if it's reliable or not.
I'm using the PFS of Nikon, and sometimes I feel it's not reaching that perfect focus, so I need to adjust manually. But I'm not sure after adjustment, I am actually hitting the best focus or I made it worse.
Yeah, I understand. I understand. And that is because you have not formulated what you are focusing on. So you have to be very clear what you're focusing on. And the reason why, if you think that the perfect focus system is sometimes not working very well, this is what I was saying, is that for a hardware autofocus, you need to have an imperfect sample. And if your sample has different refractive indices inside the sample, for whatever reason, it can be you left some PBS there, or there can be lots of different things. It can be also that you have a dish, and under the wells of the multi-well plate, for example, there's a different refractive index, so this type of things. Then sometimes the hardware autofocus gets a bit lost with this type of thing. So the higher the refractive index mismatch, and the better. And also there are some objectives that are specified for the PFS and others are not. So clearly it is important to check that the objective that you're using is actually specified to work with the PFS.
Yeah, so I was imaging the aqueous sample in an air.
With an air objective, that should work if your air objective is specified for that. But we can talk about this. Remind me, because you can also trick the software if your objective is not specified. If it's a 10x objective and it's not specified for the points, the hardware autofocus, you can trick it. So remind me this when we come to us, and then we'll tell you.
I also have a question, Sylvie.
Yes.
I don't know if I misunderstood, but I think you said that in the upright systems, these autofocus options are not available.
Only the hardware autofocus. The software autofocus is usually available.
Okay. Then I misunderstood. Yeah, because my question was gonna be, okay, how do you then define a focus strategy then? Because I have an upright system.
Yes.
Okay, but then you can do it with the software.
With the software autofocus, yeah.
Okay, perfect. Thanks.
I also have a question. In my sample, I have exactly the problem you showed with the skin sample, that like the focus is very different across the long sample. But I also do Z stacks. So for now, the strategy I had is just to choose a very large Z stack that would kind of cover the whole thickness of the sample across the different planes it is on. But that increases the amount of images I have to take, therefore, so increase the imaging time by a lot. Is there any strategy that exists to like combine maybe autofocus with like Z-stack to basically
Oh, I
shift
have
the
better.
Z-stack?
I have a better strategy.
Okay.
So you put your sample on the slide, right? And then you put the mounting medium, right? Then you put the cover slip. Is it a big cover slip?
Right now, yes, it is.
So you use a long cover slip.
Yes.
And you use a slide that has a white part. White. Some area
yes.
where, yeah. So then when you do that,
okay.
there must be some, I think someone should do a PhD about this. There must be some human behavior that pushes you to put the edge of the cover slip just to not cover the white bit. When you
Okay.
do that, the cover slip goes to the end of the slide. This is the reason why you have this problem. So it's a very simple problem to solve, and Gabby will give you the solution on Monday.
Well, you
All right.
already said, but just so if you do anything, you have your slide, you have the pluses, which in the case you actually order the correct slide for tissues. So you should always put the cover slip beyond the pluses that are marked on your slide. And then you will always be, you have your slide in the perfect position.
The
But
little
yeah, I will
X's.
visually show it to you on Monday, how it
Okay.
is.
Yes,
I'm
you
looking
see?
forward to a lecture on Monday.
Oh yeah, can you imagine all these imaging that you're going to do without the Z stack? So much better.
Yeah.
Yes.
I also have a question because it's how much difference in focus you can have for these autofocus systems to work. Because I work with plants and I have what you showed for the skin, but much more. Like in a section, I can have parts that are a micron away from the other one.
Yeah. So with a hardware, software autofocus, because it relies on imaging, as long as you can image the top and the bottom of your sample, you can use a software autofocus. Basically, the software autofocus has the same range as the objective. So as long as you can image it, you can do that with a software autofocus. But with a hardware autofocus, there is a certain range where it works. And beyond this, it doesn't work. So in general, so I wouldn't swear by that, but the hardware autofocus that I know, I think they are 100 microns. So for you, if you have 1 millimeter, you have to use software autofocus. So now the software autofocus, you can probably set it on your microscope in such a way that it does 2 rounds. So first, with big steps, so it finds more or less where the sample is, and then smaller steps around that. So quite often you can do 2 passes, let's say. So it takes time, but it can be quite nice and reliable. And again, it depends on your sample. If you have different Z planes that are in focus, then you cannot use the hardware autofocus. But if you want to, so yeah, that's a little bit the problem. So it needs to be something that is quite thin for the software autofocus to work.
But another option, if you want to keep another washboard to keep the focus, if your machine allows it, is to do a focus surface.
Yeah, like the one I gave at the third one.
For if you really have big differences and it's not like a mountain, even then you can do it, but it will take a lot of points you have to put.
It is like mountains.
But it's possible, but you have to add enough points for the software to be able to calculate, and then it will actually do it. I did it before, but I really had to, it was a huge piece of tissue, so it was like this. But I had to, instead of minimum 3, probably I added 20. But at the end, I got a very nice. It was worth the job because I slept a lot. I skipped a lot of the Z stack, so that's
because you can also
solution.
add a little Z stack to this. So you make the imaging like this, but you can also add a little stack. So it compensates for if things are slightly off. Yeah. So again, this system where you have a slanted slope and stuff like this calculation, it's a third way, and a lot of software offer this now. I don't know, different names in different companies, but you can ask the company; they will tell you.
It's really very nice way to work with.
Are there more questions? If not, let's